Hello everyone. In this video we are going to talk about parasitology, a relationship between two species where member of one species, which is smaller one parasite, derive food and shelter from member of another species large called host. Host and parasite share a dynamic relationship. Generally, parasite cannot exist independently. Three major groups of parasite infecting human beings are protozoa, helminths, arthropods. Predation is a relationship between two species where member of one species consume the member of another species for its food. Symbiosis, close living arrangement between two species, is called symbiosis. The word comes from Greek word meaning state of living together. Usually the two organisms are in close physical contact with one living on or in other. Symbiosis can be classified into mutualism, commensalism, parasitism. Mutualism is association in which both organisms benefit. In some cases, the association is so close that the two partners can be thought of a single organism. Commensalism, a harmless relationship between two species where member of one species derived food and shelter from another species. This relationship, where member of one species get benefit while other is neither helped nor harmed, is called as commensalism. Parasitism. Types of parasite. A parasite that lives inside the body of the host is called as endoparasite. Malarial parasite is an endoparasite. A parasite that lives on external surface of the host is called as ectoparasite. Sarcoptes is an ectoparasite. Obligate parasites, facultative parasites, accidental parasites. Types of host. Definitive host, multiply sexually. Intermediate host, multiply asexually. General characteristics of parasites. Human parasites vary greatly in their size and shape, however useful in their identification. Protozoal parasites are unicellular and microscopic. Their size is measured in micron. On other hand, helminths are multicellular and microscopic. Some helminths like tapeworm can measure 30 feet in length. Human parasites live in various parts of the body. Importance of studying parasite infection. Worldwide, 2/3 population is infected. Common in underdeveloped countries. Factor responsible: high population density, poor sanitation, short supply of water, poor hygiene, poverty. Life cycle: simple life cycle single host, complex life cycle two host. Plasmodium: it is a tiny protozoan responsible for malaria. Different species of Plasmodium are P. vivax, P. malaria, and P. falciparum. Life cycle of Plasmodium or malaria. When the mosquito bites the human, sporozoites are injected with bite. This sporozoites then reaches liver. Sporozoites reproduce asexually in liver. Sexual reproduction takes place in red blood cells, which are called as gametocytes. Female mosquito takes up gametocytes. Fertilization and development takes place in mosquito's intestine. Sporozoites escape from intestine and migrate to the mosquito's salivary gland. Lab diagnosis of Plasmodium. Microscopy. Light microscope is used for the identification. Uses a blood smear to identify whether parasites are present in patient's blood or not. Thick film is useful for scanning. Thin film is useful for different species identification. Rapid diagnostic test. Serological tests such as ELISA. Molecular diagnosis such as PCR. Entamoeba histolytica causes MBBS. They are unicellular organisms. It produces intestinal and extraintestinal diseases. Life cycle of Entamoeba histolytica. Mature cyst, multinucleated, injected by host. Tropocyte, multinucleated, found in small intestine. Tropocyte, mononucleated, found in large intestine. Some of these organisms invade into the organs, while some are exit out of the host. Lab diagnosis. Microscopic examination, identification of cysts and tropozoites in the stool. Serological test. Nematodes or roundworm or ascaris lumbricoides. They are non-segmented cylindrical worms. Their digestive system is complete. It consists of mouth, esophagus, intestine and anus. Sexes are separated. Male is smaller than female. From mouth of the male, it will passes to intestine, then to venous system, then in lungs, then in trachea, then in esophagus. Adult worms passes in intestine. Fertilized egg passes in the feces. Infective larva develops within egg in soil. Man ingests egg in food or soil. Lab diagnosis. Direct detection. Adult worm can be seen in stool sample with naked eyes. Larva of ascaris in sputum. Demonstration of egg in stool sample. Indirect technique. Serological test. ELISA and PCR. blood examination, imaging, X-ray and ultrasound. Glardia lamblia. It lives in duodenum and jejunum. It is found in lumen of the human small intestine. Morphology. It exists in two forms. First one is tropozoite, which is vegetative form, and the second is cyst, which is cystic form. Life cycle. Passes its life cycle in one host. Children are most affected. Ingestion of cyst through contaminated food and water. Within half an hour of ingestion, cyst hatches out into two tropozoites, multiply by binary fusion, and colonize in duodenum. Tropozoites live in duodenum and upper part of jejunum. Cysts are passed in feces and remain visible in soil and water for weeks. Passes into another human. Lab diagnosis. Stool is collected as sample for examination. Macroscopic examination. Pale colored, fatty stool, float in water and has offensive odor. Microscopic examination. Cysts and tropozoites are found. Saline and iodine wet preparation. Serological test. ELISA and PCR.